In this video, we will discuss about this beta-1 integrin and FAK's role in the proliferation in stiff matrices. In the previous video, we have seen that stiff matrices shows higher proliferation compared to the soft matrices. Soft matrices they have used is 1 kilopascal, and the stiff matrices means the polychloride gel they have used is 8 kilopascal. So first they check the expression levels of the, since beta-1 integrin and FAK plays an important role in the mechanotransduction. From that they made a hypothesis that this beta-1 integrin and FAK could also play a role in this proliferation in the stiff matrices. So first what they did is they check whether beta-1 integrins and FAK are expressing more in the stiff matrices or not. So for beta-1 integrin they used anti-CD29 antibodies, and they are able to see that for 8 kilopascals the signals is high compared to the 1 kilopascal. And this is quantitative analysis are also shown on the right hand side of this panel A. And afterwards they also checked it the critical signaling pathways including FAK pathways, and where we can see that phosphorylated FAKs and phosphorylated AKTs are expressing more in the 8 kilopascals, and along with this phosphorylated STAT3, phosphorylated ERK, and they are expressed high in the 8 kilopascals compared to the 1 kilopascal cultured cells. And these things are also quantified to show that the signals that we have seen in the Western blot are right. And thereafter, once they identified that beta-1 integrin and FAKs are overexpressed in the stiff matrices compared to the small matrices, now they wanted to see whether they plays an important role in these proliferations or not. So they one by one silenced beta-1 integrin and FAK. For beta-1 integrin, they used anti-CD29 while culturing on this stiff matrices, and they are able to see that compared to the control, that is without anti-CD29, the cell growth is low in the case where they use anti-CD29. These things are also quantified on the right hand side to confirm the results. Thereafter, they did the similar studies with a small molecule to inhibit FAK, that is PF573228, and in that case also they are able to see that the proliferation goes down compared to the control case, and these things are also quantified using this Ki67 positive cells, and we can see that Ki67 positive cells are low in the case where they use small molecule inhibitors to inhibit FAK. And then they used different approach also to confirm that they are able to see the same findings to different approach. So in this case they silence there is beta-1 integrin using silencing RNAs, and in the results are shown here for two different silencing RNAs, and compared to the control, the relative mRNA expressions of this relative mRNA expressions are low in this compared to the control siRNAs. And these things they have also quantified using the Ki67, where we can see the Ki67 positive cells are low. And similarly, so this panel E is to show this relative mRNA expression of this beta-1 integrin to confirm that the silencing RNAs are working. And then Ki67 is to show that because of the silencing beta-1 integrin RNA, they are able to see suppression in the proliferations, and they shown here using the proliferation index. And thereafter they check the relative mRNA expressions for this FAK. This FAK expressions also goes down. They checked it for two different silencing RNAs. In that case, in the proliferation assays also they are able to see the similar results, that the proliferation of the cells goes down when they use FAK silencing RNAs compared to the control silencing RNAs. And these results are also on the stiff matrices, that is hydrogel 8 kilopascal hydrogels coated with collagen. So from that these results they come to the point that this beta-1 integrin and FAK plays a role in the proliferation of the cells that can be is seen in the stiff matrices compared to the soft matrices. And if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.